Okay, so welcome to this next video in which we're discussing the Soluble Guanylate Cyclase enzyme. Okay, so far what we've discussed is the structure of the Soluble Guanylate Cyclase enzyme. We've seen that it is made up of these two subunits, an alpha subunit and a beta subunit, and that the form of the Soluble Guanylate Cyclase that is present in vascular smooth muscle cells is a form which uses the alpha-1 alpha subunit and the beta-1 beta subunit. We've also seen that each of these subunits has some important domains, one of which is this HNOx domain, standing for heme nitric oxide oxygen binding domain. Then we have the PAS regulatory domain. That should say PAS regulatory domain, not PAS regulatory subunits. So this is the PAS regulatory domain. So you have a PAS regulatory domain on each one of the two subunits, and these PAS regulatory domains bond to each other basically and hold the two subunits, the alpha and the beta subunit, together. And then finally, at the carboxyl terminal of the protein over here, you then have the catalytic domain. Right, and the catalytic domains dimerize together to make the active guanylyl cyclase enzyme. Right, okay, so that's the structure of our soluble guanylate cyclase enzyme. But we've also seen that specifically the beta-1 subunit has within its HNOx domain this histidine at position 105. Now we've drawn out the structure of histidine down here, and basically it has this lone pair of electrons which is then capable of forming a coordinate bond with the ferrous cation at the centre of a heme ring here basically. So that allows a heme ring, a phosphatic heme group, to be added on to this histidine 105 of the beta-1 subunit of the soluble guanylate cyclase. Okay, now what we want to discuss is how nitric oxide is going to actually come and activate this enzyme. Well, basically what's going to happen is, as you can see from this picture, we now have iron. This ferrous cation here has 5 coordinate bonds, but it can actually support another one basically. It can support another one coming out of the coming down here like so. And basically what's going to happen is that nitric oxide is going to come and form a bond in this way with the iron. So let's say here is our nitric oxide. It's going to come and form a bond with the ferrous cation at the centre of this phosphatic heme group on histidine 105 of the beta-1 subunit of our soluble guanylate cyclase. When that happens, it is going to activate the enzyme, and the way it's going to activate the enzyme is it's actually going to cause this bond between the histidine and the heme group to cleave. So in fact what's going to happen when nitric oxide comes in to the smooth muscle cell, it's actually going to cause all of these heme groups to fall off of the guanylate cyclase enzyme. So effectively what's going to happen is you're going to have this heme group coming off over here, and it will have the nitric oxide molecule here linked to it basically, but now it doesn't remain in this 6 coordinate bound structure over here. Instead it cleaves this coordinate bond with the nitrogen of the histidine residue and goes off with the nitric oxide, and when that heme group falls off the soluble guanylate cyclase enzyme, then the soluble guanylate cyclase enzyme is activated, and the catalytic domains which have formed this active enzyme here are going to start taking GTP in. So they're going to take guanosine triphosphate in, and they're going to convert it to cyclic GMP and pyrophosphate. Okay, so let me show you that reaction in a bit more detail on a separate page. Okay, right, so here we have another page. Right, our catalytic subunits have now been activated, and that enzyme that they form together, the dimer of the catalytic subunits, is now going to convert GTP to cyclic GMP. So let's just explore a little bit more what that means. So let's draw a little cartoon for the structure of guanosine triphosphate. So guanosine triphosphate is what's a nucleotide, basically. So it consists of a ribose sugar here, bound to the organic base guanine, okay? So here is our ribose sugar, and I should have the 5th carbon up here. So ribose is this 5-membered ring where the first member up here is oxygen, and then you have these other 5 carbons, and because obviously it's only a 5-membered ring, one of the members has to be oxygen, so one of these 5 carbons isn't going to be in the ring, and that's that one up there. Okay, so when you bind guanine to ribose, you make a molecule known as guanosine. And guanosine is what is known as a nucleoside. Okay, so a nucleoside means a ribose sugar bound to an organic base, basically. So it's different from a nucleotide. A nucleotide means that you've got phosphate groups sticking off up here. So, in order to convert guanosine to guanosine triphosphate, what you have to do is you take the guanosine molecule, so we'll copy it out here. So here's our ribose sugar, okay, with our guanine organic base off here, okay, and our 5th carbon coming up here, and then you stick 3 phosphate groups on it. So 1, 2, 3. Okay, and that now is guanosine triphosphate. Guanosine triphosphate. Okay, or GTP is what that is often abbreviated to. So GTP. So let me put some colour on these pictures so that they look nicer. Right, so we've got this guanine organic base, which we've just abbreviated to a box here in blue. Okay, because its structure is quite complicated to draw out. Okay, and then in pink we have the ribose sugar. So this is the ribose sugar here. Okay, so ribose in pink. And if you're guanosine, then that's your structure finished. You're just ribose linked with this guanine organic base. And I just want to label this up. This is the guanine organic base. So don't get confused between guanine and guanosine. Guanine is the organic base. Guanosine means the organic base guanine attached to ribose. Okay, now we've extended it even more. We've got guanosine triphosphate, which basically means we've stuck these 3 phosphate groups off the ribose, and that gives us guanosine triphosphate. Now, the reaction that the guanylate cyclase enzyme is going to catalyze is going to take in these guanosine triphosphate molecules, and it's basically going to convert them into cyclic guanosine monophosphate. And let me show you the structure of that. Okay, so if this is the ribose sugar again, and here is the guanine organic base coming off that ribose sugar, and here's the 5th carbon of the ribose sugar, then basically you take 2 of the phosphate groups off. So let's show the 2 phosphate groups that have been taken off over here. Now 2 phosphate groups joined together in that way is known as pyrophosphate, and is often abbreviated to PPI. So if you see people referring to PPI, then they mean pyrophosphate. So that's 2 phosphate molecules, well, 2 phosphate groups stuck together. Okay, and then cyclic guanosine monophosphate means that you've now only got 1 phosphate, but the cyclic means that you've bound the other end of this phosphate that was bound to a 2nd phosphate up here to the 3rd carbon of the ribose sugar, like so. Okay, so what you end up with is this cycle here, and that's why it's called cyclic guanosine monophosphate. Okay, so let me colour these bits in. So in blue we have the guanine organic base, and in green we then have our final and only phosphate group left on, and this basically is cyclic guanosine monophosphate. So cyclic guanosine monophosphate. So that's the reaction that this soluble guanylate cyclase is now going to catalyze. And cyclic guanosine monophosphate is often usually abbreviated to cyclic GMP. Right, so when the nitric oxide binds to that phosphatic heme group, it's going to cause the phosphatic heme group to cleave off of that histidine 105 of our H-NOx domain of our beta-1 subunit of our soluble guanylate cyclase, and that means that this catalytic portion of the enzyme right at the base is going to become active, and it's going to start taking in GTP and breaking it down into pyrophosphate and then a cyclic GMP molecule. Okay, right, so in the next video what we'll do is we'll look at pharmacological agents which we can use to interact with the soluble guanylyl cyclase.